the first time we're looking at abiotic stress in the BGI, so that's fantastic. And I wanted to give a little bit of background in relation to that. So these are the, which one's the pointer? That's the pointer, right? These are the scientists in the wheat physiology group, Carolina Rivera. She's in charge of data automation and management, a new addition to the lab. Also Francisco, he liaises with the breeders within CIMMYT and also in the Mexican National Program, since we get money from Mexico, and he's also an expert on lodging. Jacinta Jimeno, she is the IWIP Hub Manager, that's the International Wheat Yield Partnership, and she has, most of her work is related to logistics of a large number of collaborators who work with us in the platform in Mexico. Francisco Pinto, he gets to play with the toys. He's in charge of high throughput phenotyping, and he's also helping Bread Wheat with their genomic selection project. And then there's Siva Kumar Sukumaran, who's leading the pre-breeding and molecular breeding. Gemma Molero, who's been there the longest after me. She's a photosynthesis expert and also partitioning and essentially leads the training of the PhD group. Carolina St. Pierre, who's here in the audience, I think. Yeah, there she is. She also helps us. She's leading the phenotyping hubs, the international phenotyping hubs, and transgenic work. Many collaborators, too many to go into, but from all continents, as you can see. And of course, a complex mosaic of funding, including several public-private partnerships. So that's a little bit of background before I get into the talk. So what are the objectives of wheat physiology at CIMMYT? Well, first of all, we need to obtain proofs of concept for the physiological traits and alleles that can boost yield in a realistic breeding context. And if you think about it, if you look at the literature over many decades, there are literally thousands of hypotheses about traits, physiological traits, photosynthesis, roots, whatever, that can boost yield. But so few of them have really been tested in a breeding context. So in some ways, that is the most important objective. And then also to broaden the gene pool through selective use of genetic resources. And I underline selective because that's exactly where physiology can help. There are many genetic resources, I'll come to that, in the world, but we need a basis to be able to pick the most promising ones, and that's where phenotyping and particularly high throughput can come in. And then, of course, to deliver novel sources of traits in acceptable agronomic backgrounds to breeders worldwide, including our own breeders, obviously. So that's where the pre-breeding pipeline comes in that I'll describe. And lastly, and certainly not least, provide crossing and selection protocols that can complement mainstream breeding. The pre-breeding we do is very much, the point of it is to complement conventional breeding. And with that in mind, all of our protocols are shared. And for example, in these manuals that have been very widely distributed and translated into Chinese, Spanish, Alex just told me Russian next, Persian, they're being translated into Persian at the moment, and a number of publications that talk about the concepts of physiological breeding or pre-breeding, and of course some proof of concept for raising yield potential was recently published. So just as an outline, I'm going to describe the pipeline very briefly. Obviously we have to start off with, I guess this pointer works both ways. That's what I was, oh no, that's not the pointer. This is the pointer, yeah. So we start off with crop design. As I said, the literature has many ideas. You put together a set of traits that you think would improve adaptation to a particular environment. And then you need to look for good sources of those traits, and that's where we go to the genetic resources. Again, the phenotyping is super important there. Genetic analysis helps us with understanding which traits may be complementary or where there may be trade-offs, as well as genomic selection, and then of course the crossing and selection, and finally the evaluation in important target environments. So those are the key steps that I'll go through. So starting with the first one, identifying crop characteristics that confer adaptation. Here's a set of traits that we essentially can confer heat adaptation. Don't have time to go through all of them. Would love to. Obviously, the point I want to make here, first of all, is that we develop these conceptual models that serve as platforms for both research and pre-breeding, but also that there is always an interaction of any trait with a genetic background. If there weren't, breeding would be easy, and it's not. And of course, there's always an interaction with the environment. And just by way of an example, here we see a set of genotypes that were selected from a pre-breeding cross or set of crosses, and these were grown in two different drought environments. One that simulated a Mediterranean type of drought, so drip irrigation, another that simulates a post-monsoon type of drought where you have a full soil profile. Exactly the same genotype, same sowing date, same year. And what you notice is the close to zero correlation between those two environments. That's G by E. Very low correlation. So, second point, exploring genetic resources for adaptive traits. Well, briefly, as you can see from the photo, this is very much a bottom-up approach to genetic resource screening in more senses than one. It's very basic because there's not a lot you can measure on 70,000 genetic resources. That's the number we screened, about half of the world wheat collection in a collaboration with Seeds of Discovery. There's 0.8 million genetic resources in the world, wheat genetic resources, estimated number. So there's a lot of opportunities. And what you do with this is, here's an aerial photo of that kind of sowing. The idea is to pull out panels of diverse but promising material so that those can be used for more detailed characterization for both use as parents in pre-breeding as well as for genetic studies and other kinds of research. And here's one such panel, bred wheat diversity panel, again consisting of land races, lines derived from interspecific hybridization, elite material, and so on. Here's another panel. These are primary synthetics. We've heard about synthetics today. These were selected from 2,000 that were generated mainly by Mujib Qazi at CIMMYT over the decades. And this is a set that shows a lot of variation and some excellent adaptation to heat and drought and also yield potential. I'd love to tell that story, but not enough time. So high throughput phenotyping. First of all, it's important to remember that phenotyping is not just about tools, even though that's what we tend to get excited about. And this is an example of two sister lines. Because of the hexaploid nature of bread wheat, two sister lines can have this kind of difference in phenology. And these would just happen to be sitting next to each other in a population, in a double haploid population. And problem with that is, how do you phenotype it? One line is about to reach physiological maturity, while the other line is about to head. Essentially, these are two lines growing in completely different environments just because of their phenology. And so we put a lot of effort into developing panels like this, like the Seri-Babax population, where the phenology is well controlled. Also, much easier with an association panel you can deliberately control. Essentially controlling the genes of major effect, because typically complex traits by definition are made of genes of minor effect, and it's very hard to work with those if you've got major genes confounding your population. Okay, so what are the tools? These are some of the high throughput tools. Here's my favorite question for my students. What is the best physiological tool available to any plant breeder? And I won't wait for an answer, I'll just go ahead and tell you. It is, of course, the low-resolution stereoscopic spectral radiometer connected to the supercomputer. And if you haven't figured that out, that is your eyes and your brain. However, so even though I'm a physiologist, I insist that that is the best physiological tool available to a breeder. However, the eyes and the brain together, they do not see certain things. They do not see temperature. And temperature is extremely well associated with yield and other characteristics under stress, for example. And also our eyes don't have the resolution. No, they say low resolution. Our eyes will see green, but we won't see embedded within the greenness of a leaf, pigments like carotenoids. We only see those in the autumn when the chlorophyll is degraded. A spectral radiometer, on the other hand, can pick out those details with great precision, every nanometer of light, and give you a number. And so we need tools to help us see better, essentially. This graph shows the association between grain yield and canopy temperature. Typically, a very high association can be seen, explaining, this is confounded in this case by environment, but even within environments, you'll explain 50% of the genetic variation just with this one trait of canopy temperature. Not under high yield conditions, but certainly under stress. A very valuable tool, easy to use, almost foolproof, very inexpensive, very robust. And one can easily teach a technician with a very limited education to use this very effectively. One of the other great things about canopy temperature is shown in this graph. What we're seeing here is the association between deep roots, between 60 and 120 centimeters, so almost in the subsoil, and yield on the one side, so a linear relationship, and canopy temperature on the other. So the cooler the canopy temperature associated with deeper roots. And when you think about the effort that is required to measure roots, especially roots, as several people have mentioned today, shovelomics, and they're talking there, quite hard work about looking at superficial roots, crown roots often, or the superficial seminal roots. But here we're talking about roots below a half a meter. These are very difficult to dig up. You need a lot of hardware, tractors, and so on. And yet, a simple and easy to use trait like canopy temperature is explaining in this set of sister lines almost 90% of that variation. And so this is a real opportunity to be able to measure a very difficult trait with a very simple tool. And we're pursuing this at the moment, looking at other environments other than drought. But of course, we can go to the air, and that's what everyone wants to do now, fly drones. And that's exciting. There are opportunities to clean up data. We typically see better associations with performance when you're looking at aerial data. Partly that's because you can clean up the data, as you can see in this histogram. You can remove outlying pixels with image analysis. There's all kinds of reasons why you get variation within a plot. You can almost see it in that figure. And you can also take a snapshot across many plots, and that avoids the environmental effects of running or walking rather between plots with a handheld instrument. So there are advantages. There are disadvantages too, because image analysis is not a straightforward thing. It typically requires a lot of expertise, but no time to go into that right now. Other kinds of opportunities are, for example, here we see 3D reconstruction of this, not a photo, that's a 3D reconstruction showing plant height. And what we would hope to do with this is to find ways, for example, to estimate biomass. Biomass, such an important trait, so rarely used at a breeding scale because it's not that straightforward. But with combinations of traits like, well, for example, the ability to reconstruct, to calculate the volume of a plot and perhaps including water index, we are probably able to, this is what we're testing, the ability to estimate in-season biomass or even final biomass more precisely than we can at the moment. So the genomics, or as I said, useful to help you design crosses. and also looking at trade-offs between traits complementarity, only the alleles. Roberto always tells me this, only with the QTLs can you really be sure of what you're talking about, and I completely agree. And of course, genomic selection. So just a list of some of the molecular marker studies led by our group for traits like canopy temperature, earliness per se, grain weight, adaptation to agronomic density, fascinating subject, yield and components, height, spike photosynthesis, I'll come to that one in a minute. Lodging traits, another key one, spike ethylene under heat stress related to progeny load, and other QTL hotspots really in the durum that we've looked at. That's a few of the studies we've conducted. No time to go into details, except for this one. I thought it would be nice to talk very briefly about spike photosynthesis. Why? Because in this picture, you can see very clearly how much of the light that is intercepted by spikes. A very large proportion of the incident radiation is intercepted by spikes, and yet we know very little about their photosynthesis. And it's a very difficult trait to measure as well. Typically use gas exchange. What we did in this case was to estimate it indirectly by blocking the light to the spikes, so we could estimate the contribution of spike photosynthesis by subtraction. And with that information, because that's a much more high throughput approach than gas analysis, we're able to show the first QDLs for spike photosynthesis. So that's just an example of something, an application where a difficult to phenotype trait could be overcome by, if we can develop markers. Genomic selection, we're using our pre-breeding material to develop genomic selection models so that we have more confidence in the lines that we send out to international nurseries. And of course, the predictions, as you would expect, as you can see in this figure, they're much better for the heritable traits than they are for the less heritable traits like yield. So finally, what all this leads to is strategic crossing to accumulate genes to improve yield. And I'll give you an example. This is a hypothesis that we wanted to test, the idea that if you get a better balance between the source, in other words, the photosynthesis, and the sink, which is the partitioning to yield, that one can increase genetic gains. And I won't go into all of the traits, but these are the traits that we use to select parents to make these complementary crosses. And as you can see, they're categorized as source or sink. And the progeny, to cut a long story short, of those crosses is selected within the IWIN at a subset of sites within the IWIN. And what we see, these are some results from 32 sites and some low G by E clusters. What you can see is some marginal gains over the elite check, Borlaug. That's about a 4%. So sometimes significant, marginal, let's say. But what is much more exciting, I think, about this as a proof of concept for pre-breeding and the use of complex traits that can be adopted in mainstream breeding is that although the yield is quite marginal, the yield gains, and in some environments substantial, in others less, but the biomass very consistently higher. That's a 25% increase in biomass, so a huge jump in radiation use efficiency. And that is exactly an outcome you would expect and want from a pre-breeding program, because this material is not intended for release, it's intended for use by breeders with a 25% increase in radiation use efficiency, which is then crossed and locally adapted. One can expect that a significant proportion of that increase can contribute to increases in yield. The other exciting point about this is that all of the lines that beat the check, the CIMID check, they all show exotics in their pedigree. So many of them have both a landrace and a synthetic in their pedigree, some just a synthetic, some just a landrace. Essentially turning over that idea that at least for complex traits, that the exotics slow you down. We didn't find that. By selecting well, the exotic, we're able to achieve this kind of progress. So here's a field of wheat called Pakistan 13 in Pakistan. This is what can be achieved when breeders and physiologists work together. When the physiology is integrated with the breeding, these are all lines coming from exactly the same kind of approach. And they've all been released in Pakistan over recent years. And lastly, it's important to remember the next generation. And I just wanted to show you a photo of some of the students that we've trained in recent years. These guys, except for Hemmer in the middle, have finished their PhD by the end of last year, different universities around the world. And this is our new crop of PhD students who are studying in different universities, many of them supported by generous support from the Mexican Science Ministry. So that's all. Thanks very much.